Okay, hi everyone. Thanks for joining. So we're continuing with our series about resources for research, exploring or bringing to you guys some information about our fantastic HICCC shared resources. This week we have two. The first one is the genetically modified mouse models resource that'll be presented by Jean-Wen Kay, who's the director of that resource. And our second one, I'll introduce that when Jean-Wen finishes. So go ahead, Jean-Wen.
Okay, thank you very much for that. I hope that you can hear me then. Okay.
Yes.
Good. So I'm a director of this genetic modified mouse model resource, and so my the short name for our core is called GMMMSR. We have Victor Lim as the technical director, and then we also have a Hui Chum as a microinjection specialist. And both of them have many years of experience, especially Victor, who is also associate professor in Columbia University, have a very strong research experience and also mouse design experience as well. And so we have received support from Cancer Center, but also we also generate large avenues from our calls to supplement our salaries and also expenses as well. And our GMMMSR has a very strong advisory team, including Sanja and Corey and also Laura. They're all fantastic scientists in Columbia University with a strong experience with this cancer research and mouse model design. And our core essentially provide three ways of making mouse lines, including conventional transgenic mouse lines, including using this DNA construct, deliver this construct into fertilized mouse eggs. And we also have this, the second one will be more prominent and also more popular tool and ways to make mouse lines using this ESL-based gene targeting strategy. We knocking knockout mouse lines and also using making pre-ER mouse lines that many labs are using. And essentially, I will give more introduction in the next few slides about this technology, but we use this engineered embryonic stem cells to deliver them into blastocysts to make mouse lines. And then, of course, then very become more and more popular is the third one would be this genome editing service using CRISPR-Cas9 system. So with this conventional transgenic mouse lines, as I briefly mentioned to you, is that you could deliver DNA, inject DNA into fertilize the eggs for a nucleus, and then implant these eggs into pseudo pregnant mouse, and which this pregnant mouse then will give birth to pups, and you could then using a DNA analysis to find out these founders and then propagate your mouse lines of interest and using PCR technique. And a more popular strategy, as I mentioned to you, would be this ESL transgenic targeting system. And here involved is making gene targeting construct and to replace the endogenous gene with engineered allele with homologous recombination. And by that way, you could do a deletion, point mutation, making a flux alleles, also making mouse lines with the GFP, RFP, and M-cherry. And then you would do these gene manipulations in cultured embryonic stem cells and using drug selection to select those cells carry your gene of your interest, and then we propagate those gene of interest clones, and those are the target ESLs that you want to for the next injection into the blastocyst. And similarly, then you can transfer this blastocyst to mouse, pseudo-pregnant mouse line to create mutant mouse lines. And here, this is the cartoon that you can see. Initially, we make these ESL lines by Victor, and he take out this inner cell mass and put in tissue culture to make ESL lines. And we have made a few bona fide cell lines, including in black 6 genetic background and also 1 to 9 genetic background as well. And then once you have these ESLs, and then you could transfer your target into the ESLs using electroporation, and then followed by drug selection to select the clones that you are interested carrying the gene of your interest, and then um, um, these are the clones that you want to expand, and then you can deliver that into the blastocyst. Then the blastocyst carried of your gene, carry the ESLs now will be, which carry your gene of interest will be transferred into pseudo-pregnant mouse mother, and then they will carry pups and eventually give birth to these chimeras. And that chimera will a mixture of your host and also the ESL that put in. Then you can breed these chimeras with wild type mice and to separate your gene, the offspring that carry the ESLs of your interest. And those are your mice, and we will provide all this service, and which I will talk about later on. And then of course, then we go with a trend. We also offer this CRISPR-Cas9 mediated mouse clone strategies using this non-homologous anti-joining system, and could using guide RNA and put into a single cell embryos along with the construct that you are interested in, and then making these mouse lines similarly put into these pseudo-pregnant females. And our core provide this different type of strategy to making mouse lines, and essentially we want to provide and make this very convenient to our users. And for example, we now help users to generate targeting vectors, and we could use, for example, DNA synthesis, and also do a gene targeting for you as well. We could do very systematic way, comprehensive way for you, help you to generate mouse line, which we call full gene targeting service, which include the service from design of the construct, vector construction, and until germline transmission, and the whole duration takes about 10 months. And we also use CRISPR gene editing in ESLs and embryos. And I just want to say that our core assisting our clients with to design experimental approaches, and also provide technique consultation and troubleshooting. And we also try our best to keep up with all the new technology to make these transgenic mouse lines. And we also constantly improve our efficiency as well. And with these high quality ESLs, with robust Black 6 ESL clones for gene targeting service. And with this full gene targeting service will allow our users to generate these novel mouse lines within 10 months normally, but we have these 12 months backed by full refund guarantee. For example, if you want us to help you with the mouse lines, and 10 months later we don't get the mouse line for you, and we would 12 months later, if we don't get the mouse line for you, then we will refund you the money. And we also take orders from other institutes and universities as well. So if you have collaborators or friends who are interested in using our facility, our service, and please also contact us as well, and we would give them a good deal for making mouse lines that of their choice. And in addition to directly provide service for making mouse limes, we also provide this consultation and training for grant purpose. And we also provide education and training for your for your for your trainees, for example, and postdocs and graduate students. We could give them education, give them knowledge that we know about how to make mouse lines. And of course, we want to make this standard rigor and reproducibilities. This is very important for the mouse lines. You don't want in the end to get mouse lines that doesn't carry your gene of interest. So we held very strong standard for that, because we don't want in the end waste your time, waste your money, and also waste our time as well. So this is the mutual interest that we really cherished. And with the new technology coming into the field, we will also learn by ourselves as well. And once we learn, and then we will teach our users how to use those new technology to make mouse limes and manipulate embryos as well. And this is just a list of the fees for our service. As you can see, they are different type of service that we provided, and we have a rate for our members, cancer center members, and also non-members in Columbia University. And for the external rate, which is for those who come from other institute, the price is a little bit higher. But this I just want to say this is not fixed. We are a little bit we could be more diplomatic in some way, but we just want to provide the best service for our users, not only just in our Columbia University, but also from universities outside of in other universities as well. And here I just want to point out about 16. You can see this is full gene targeting service. For our center member will be about around $20,000, and for internal is about $25,000. And this is very good deal if you think about the time that you put in design of a construct and making ESLs and also injections and waiting for breeding. Since takes time, I would suggest you, if you have something that gene that you are interested in your mind, this would be one of a very good deal you should take. This for gene targeted service. We not only offer making these mouse limes with this just mouse lines, we also give you a targeted genes in 1 to 9 using 1 to 9 ESLs or B6 ESLs, which sometimes will be particularly important for your research in terms of mouse genetic background. And we also accustomed to those requests. And this is just, again, to talk about this full gene targeting service. You can see the timeline, about 1 month for vector construction, and then 1 month for targeting in ESLs, and followed by a few other 1 month or 2 months time with these breedings and injections, and eventually take 3 to 4 months for germline transmissions. And our research has been used many published by many papers in these years, and you can see some of this published in very high quality journals like impact factor larger than 20, Nature and Cell, which shown here, for example. And you can see that Sanja published paper with our mouse limes in Nature, and very recently Kelly published her paper in Cell, also using our facility to generate mouse lines for her research, and myself using our core to generate mouse lines as well. We have a paper published in Nature too. And team just published a paper in Nature, also used our service to make the mouse lines. And you can see that maybe you could be one of the next high profile publications by using our service. So again, we are very flexible, and we are also knowledgeable for making mouse lines, and um, I particularly want to emphasize this full gene targeted service if you are interested. And we have a very good quality control, easy communication, and again, that you don't need to get this transport in quarantine. We know that if you get these mouse lines or generate mouse lines with other companies or other calls, then those mouse lines that you generate need to be quarantined. And if you use our call, then you don't have those trouble, and that saves you a lot of time. And this is a contact for our call, and you can always email me. And this is my phone number as well. I just don't always be with my phone close to my phone, but would be good to just shoot me by email, and also Victor email and Victor's phone. And if you interested in face-to-face talk, and Victor and myself will be more than happy to talking to you. And we are in Cancer Center in sixth floor. And with that, I would take any questions.